speaker is Gerrit Borchard, and I see that we really are behind schedule, so I'm not so sure that our questions and debate session will last very long or will be there. Gerrit is Professor of Biopharmaceutical Sciences, President of the Swiss Society of Pharmaceutical Sciences, and works in Geneva and Lausanne, so in this non-European country. Or did I say something wrong here?
Yeah, but only details. Good morning, everybody. Be happy to be here. Just a curious thing, I just realized that in 2 years' time, the very country where EMA is currently located will be a third country as well. Interesting. So, I'm not only a scientist at the University of Geneva at the School of Pharmaceutical Sciences, Geneva, Lausanne, but I'm also a trained pharmacist. And from that point of view, I'm not only involved in projects having to do with nano that we run at our group at the University of Geneva, but I'm also a trained pharmacist. And from that point of view, I'm interested in what happens if these concepts that we're working on in my lab then reach a certain stage where they can be released into the market. So this is working with the compendial or at compendial issues is certainly a purely pharmaceutical activity, as I would say. Now talking about pharmacopoeias, just as an introduction, you have these three major pharmacopoeias in the world which do have inherently different structures. The Japanese Pharmacopoeia is a governmental organization under the leadership of the Ministry of Health, Labor and Welfare in Japan. The US Pharmacopoeia, we'll talk a little bit about this, is a not-for-profit organization. It was created before 1900, but its incorporation was in 1900, so a pretty old organization. Not-for-profits are not under the leadership of a governmental institution. Whereas the European Pharmacopoeia is under control of the EDQM, the European Directorate for the Quality of Medicines and Healthcare. It's a Council of Europe institution, and this is intergovernmental, so there are a lot of countries in Europe involved in this, including the other European countries like Switzerland. So, first of all, going to the EDQM and the European Pharmacopoeia. It's, as I said, a Council of Europe directorate. It's based on the convention, elaboration of a European Pharmacopoeia in 1964. It's a basic contribution, a mission to contribute to a basic human right, access to quality medicines and healthcare. Then in 1994, the European Pharmacopoeia Convention was signed, and creation of procedure, certification, suitability to the monographs, 2007, 2009, several activities were transferred to the working load of EDQM and the European Pharmacopoeia, as you see here. Now, the mission is to protect public health. The European Pharmacopoeia is an official pharmacopoeia for the member states of the convention, which means that this is mandatory at the same date in the 37 member states of the Council of Europe and the European Union by a decision of the European Pharmacopoeia. So it's binding. However, there are certain monographs and different national pharmacopoeias that are only applying to certain countries, so they are in addition to the European Pharmacopoeia. It's legally binding quality standards described in the Pharmacopoeia and monographs and so forth, and there is a harmonization, global harmonization between the USP and the Japanese Pharmacopoeia in order to save time and money for everybody. Now, how does this work? How are monographs made? And there's a little difference between how this works at EDQM, or the European Pharmacopoeia, and the US Pharmacopoeia. Well, first of all, there is a proposal being submitted to the European Pharmacopoeia Commission, to come by a stakeholder, which would typically be a national organization. For Switzerland, for this country, this would be Swissmedic, who approaches the European Pharmacopoeia with a suggestion to put onto his program the development of a new monograph, for instance. And the COM decides to elaborate or revise an existing monograph. So this would be a new monography. This would be a revision of a monography. And then it gives that, if they decide positively, to a group of experts or to a working party, which sounds much nicer than it actually is. Then a rapporteur from this working party or from this group of experts then prepares a draft monograph, which is then evaluated by this group of experts. Again, it goes into publication in Pharmacopoeia. It's a draft monograph which is published for inquiry by several stakeholders, involved industries, by medical agencies and so forth. And this is a publication for 3 months in Pharmacopoeia, which you see here. It's the official organ of the European Pharmacopoeia, where it's online, published 4 times per year, and you can look at this even online. Now, then there were comments are being made to this draft of a monograph, which go back to the EDQM by these national pharmacopoeia authorities, which will process the comments received on the graph, and this goes back to EDQM in order to be talked about, discussed in the group of experts or these working parties. And finally, there might be a revision of the monograph according to the comments received of these 3 months of evaluation and comment phase. And then this would go back to the European Commission, which then would adopt this monograph if everything went well, if necessary with slight modifications, and then there's an implementation date, which is 1 year after the adoption of this monograph. Yes, things take time, even at the European Pharmacopoeia. There's a revision, so this is not carved in stone, this monograph, but there's a revision like 3 times a year, which can then still revise whether the monograph is then revised or adapted to the current situation. I'm part of this non-biological complexes working party. So that shows that not only the regulatory authorities like FDA or EMA are involved in this discussion around non-biological complex drugs, but it was also seen by the European Pharmacopoeia, and as I will show also by the USP, that there's a need to discuss these type of drugs, if you want. This was created already in 2011. I came to this group in 2012 or 2013 on an initiative by Swissmedic. So Swissmedic proposed to the European Pharmacopoeia Commission to install this working party on non-biological complexes. And then the European Commission decided to take on to its work program, a first example of such NBCDs. It's iron sucrose concentrated solution. Now the funny thing for me is that we are talking here about products that have been released onto the market since a long time already. And now we're setting, if you want, quality attributes or critical quality attributes in the monograph that have been already identified by the producers of manufacturers of these drugs. So then we're tasked with the elaboration of a monograph for this, and this should be used as a template, a first template for other such drugs in terms of the NBCDs. So just a quick slide on the iron sucrose drug products, why these we think is rather a complex drug. What you have here basically is a iron oxohydroxide core, which is coated more or less with sucrose, with a carbohydrate. There are different forms of that. There are also different carbohydrates used in order to coat these iron cores. In the central and the core is a polynuclear material. The differences in core size, in the carbohydrate chemistry, in the nanoparticle characteristics, and for us it is interesting, what are these characteristics? What are the things we need to measure to put down as tests in a monograph? Because these nanoparticle characteristics will have an effect on the PK, the PD, safety, immunogenicity, because these nanoparticles are interacting with the immune system, so all these properties should play a role and should be characterized properly. Last thing is that the stability of the polymeric iron complex is also interesting, because what you have in the production is that there may be some of these ions, iron ions, being released from the core, from this particle, so-called labile iron, which may cause some side effects of this, this drug, leading to efficacy or impact on the efficacy and tolerance of these drugs. Now, what we have here is already in the USP and the BP. There were some monographs citing iron sucrose injection in BP and USP, and this is sort of a draft. Just as a comparison, we looked at what has been written in the USP, what has been done in the BP, and then we discussed internally in this working party what we have to do differently, maybe based on the current progress of scientific knowledge, and what we should add as tests which would be important for the quality of this drug. This is the current composition of this working party. You see that we have people from academia, from the universities. We have people from the producers and manufacturers here. And we have representatives from the regulatory authorities in Germany as well as in Spain. And these are rather vivid discussions. I'm heading out tomorrow to another meeting, face-to-face meeting to Strasbourg to discuss this further. Now, this was on the on the European Pharmacopoeia. How does this work for the USP? There, the USP is approached by a sponsor. That could be a manufacturer of an originator product, of a generic or similar product. This is reviewed by USP staff, the Council of Experts for review. This Council of Experts then gives approval to a publication, a pharmaceutical forum, again for public comments, like we do in the European Pharmacopoeia. And the expert committee then reviews these comments in response to USP scientific liaison. Then this liaison office compiles and analyzes these comments and response. So either they think that a further revision is needed, so it goes around again, or no further revision is needed, and then it becomes an official revision published annually in the USP NF. And of course, there's also a feedback mechanism where these monographs then are regularly reviewed and adapted as necessary. Now, giving you a flavor of other NBCDs, we've been talking a bit about glutaramers. The story behind this is that in the Weizmann Institute in the 1960s, people tried to set up an animal model for multiple sclerosis called the experimental autoimmune encephalitis, or EAE. Now, you do this by chopping up this protein that's insulating these axons of the nerves into its components, which are basically these 4 amino acids. And then you combine these amino acids into polypeptides. And then you use the synthetic copolymers that you receive here by synthesis. You inject into these poor animals. And that was supposed to induce EAE, so an autoimmune response to these polypeptides. What happened, however, these guinea pigs did not develop EAE. Quite on the contrast, they were protected against EAE, and they lived very happily forever after that. So Teva, who was then bringing this forward, this product forward, then produced, based on these experiments, make a long story short, the drug Copaxone, which is still in use in MS patients. However, what they found is that these polypeptides that have been produced may be a number of 10 to the 26 different structures, so a very complex drug. Just to give you a reference, this is more, 10 to the 26 is more than there are stars in the known universe, as far as we know, so very complex. The mechanism of mechanism of activity, we have some idea, or people have some idea about that, but the actual proof is still to be seen. Because there are so many different peptide structures, you cannot establish a clear PK/PD profile in this case, because you don't know which of these peptides you have to follow. Now, in 2015, FDA approved a generic of Copaxone, that's the Sandoz and Momentus Galtopa. And based on this, in the aftermath, I think, of all this, the USP installed an expert panel for glutaramers to develop a monograph with the participation of all the stakeholders that are involved in this. And recent development was in the last, I think it was the last meeting in Strasbourg. The EDQM also decided to take on the development of a glatiramer monograph onto its program, and that's been given to a group at the EDQM which is involved in peptide analysis. As I said, there is a harmonization between the different pharmacopoeias in the pharmacopoeial discussion group, which was set up in 1990. I think they meet twice a year, which drives this international harmonization of the different pharmacopoeial requirements into a single set of global specifications in order to avoid redundancy in testing, so to have a harmonization in the testing, reduce overall costs for the stakeholders, and reduce time to bring this to the market. Harmonization in parallel and coordination also with the ICH, for instance, and other interested organizations. So I hope I could give you a short overview on what's happening in the compendial side of this, and I think for this rather complex topic, we need all the king's horses and all the king's men to come to a clear picture of this. Thank you very much.
Thanks, Gerrit. Any questions? Yes, please.
Can you press the button? There's a button somewhere.
Can you hear me?
Yes.
Sorry. I was very interested, I'm Sarah Michelle from the University of Maryland, and we're actually we're going to be doing an FDA-funded clinical trial of the iron gluconate drugs, the branded generic. And I was very interested in this idea about the labile iron in the drug to begin with. And I don't know if you or perhaps Stefan could comment on this. Do you believe there is labile iron in the actual drug? And then what's a good way to measure that?
Yes.
We've been grappling with that in my lab.
Yeah. Okay. You have the resident expert for labile iron sitting here in front. It's a student of mine, Tiziana Francesco. We've developed or tested several assays to measure labile iron in these formulations. We ran through 7 different assays, and we found 2 to 3 which are giving us reproducible data. We think that one of these is giving us too low concentrations based on what information we got from the different producers, manufacturers. The other one is a little too aggressive, so we think that this is actually attacking the complex and then releasing, artificially releasing the iron. Now we got a third one, which we obtained from Sigma Aldrich, where you can measure at the same time iron 3 and iron 2.
Okay.
And we are on, Sigma would not reveal the composition of this test, but we are on it, so we probably have an idea of doing this. So making a long story short, we think, yes, there is, first of all, labile iron, which may cause some side effects there, and we have some pretty good idea how to measure this, at least in vitro, so in the assays we have.
And so they use chelators? Is that sort of what you're...
Yeah.
Okay, so you just sort of match it to what you predict the KD could be.
Yes.
Okay.
Yes. And we're about to publish that also in the journal,
terrific.
but Tiziana will be available for discussion.
Great.